So we've talked a fair bit now about the defenses that the body has that work against all potential invaders. Now it's time to talk a little bit about how the body can learn to respond in a very aggressive way to very specific invaders. So we're now ready to talk about specific defense or adaptive defenses and what we term as immunity. So to get started, the adaptive defenses or immunity is provided by the actions of lymphocytes and macrophages in response to the presence of specific antigens. And so let's introduce first the lymphocytes that are involved. The first set of lymphocytes are the T cells, also known as the T lymphocytes. And I think about them as being T, that T is telling us that they are thymus-derived. So, like all of the formed elements, the T cells are made in the red bone marrow, but before they are fully developed, they migrate, they leave the red bone marrow, and they migrate to the thymus, and there they complete their development. So, it's under the direction of the thymus and the thymosins, these hormones that are produced by the thymus, that the T cells are trained. So they do the last of their development there in the thymus, and then once fully developed, they take up residence in the lymphatic organs, and we find them especially in lymph nodes. There's large masses of them in the thoracic duct and also in the spleen. The other kind of lymphocyte that is important in immunity are the B cells, or the B lymphocytes. But rather than requiring migration to the thymus to complete their development, they become fully differentiated in the red bone marrow. So they do all of their growing up and training there in the bone marrow. They also, once they are fully developed, they also settle out in lymphatic organs, especially the lymph nodes and the spleen, but many of them remain in the bone marrow, and others, again, masses of them can be found in the intestinal lining as well. So slightly different locations, but still, that and the fact that they're not, that a location isn't an especially location, that doesn't mean that it doesn't have T cells and B cells living, taking up residence there. These are just the places where they are the most abundant. So, what is it that they do? Well, the first thing that has to happen before they can learn to recognize these specific threats, we've got to get them activated. And so we'll start first by talking about T cells and T cell activation. So, a T cell is trained to recognize a specific antigen when it comes into contact with that antigen on the surface of what is termed an antigen-presenting cell. So these antigen-presenting cells, they can be macrophages, they may be dendritic cells, and they can also be B cells. These antigen-presenting cells will phagocytize some kind of antigen-bearing particle. So it could be a virus, the virus itself. It could be that it phagocytizes a bacterium. But when it does, rather than fully digesting that antigen that's there on the surface of the antigen-bearing particle, it instead leaves the antigen whole and takes and puts it out onto its own cell membrane. So it presents the antigen that it's collected off of whatever the pathogen is. And it displays the antigen near another group of proteins that are called the major histocompatibility complex proteins, the MHC proteins. So it displays it near this major histocompatibility complex, and that's what helps the T cell to recognize the antigen and to recognize that it needs to respond to it as a foreign element. So the antigen-presenting cell comes into contact with the T cell. They interact. The T cell binds the antigen, and it also binds associated proteins on the antigen-presenting cell's membrane. So if it doesn't get both of those things, these associated proteins and the antigen bound to the T cell doesn't become activated. But if it does bind that protein, bind the antigen and the associated proteins that are nearby, then the T cell is activated to recognize that antigen and that antigen only. So when a T cell has been activated and it stumbles across an antigen or one of the particles that's bearing that antigen, these are the things that it can do. The first thing is that it secretes these substances called cytokines, and they enhance cellular response to antigens. And so there are 4 cytokines that it can secrete. It can produce interleukin-1. That is a substance that helps activate other T cells and stimulates them to produce cytokines. Interleukin-2 causes T cells to reproduce and stimulates them to produce more cytokines. So interleukin-1 and 2 are going to get more of these T cells out there that have the capacity to recognize the antigen. It secretes chemicals called colony-stimulating factors. These are, and we talked about these back when we talked about the formed elements. These colony-stimulating factors help to stimulate the development of leukocytes in red bone marrow. And then finally, they will secrete tumor necrosis factor, which helps to stop tumor growth. It can stimulate the growth and differentiation of lymphocytes so that we get more fighters. And it can also act as a pyrogen. Remember, pyrogens were those chemicals that talked to the hypothalamus and told the hypothalamus to raise body temperature. So it will secrete all 4 of these cytokines that are going to help to get more cells responding to the antigen-bearing particle. It will also interact directly with the antigen or the particles and secrete toxins on them. So these toxins include perforins that are going to lyse the cell and kill it. It can secrete growth inhibiting factors that are going to slow the growth specifically of the target cells, the things that are bearing the antigen molecules. And it can secrete these substances called interferons that are going to slow the growth of virus and tumor cells. So we secrete the cytokines that's going to recruit more of the T cells to come and help do the fight. And then it will go out and fight off the antigen-bearing particles by killing them, by inhibiting their growth, and by producing interferons that are going to slow down the growth of other cells. And so we talk about the activity of T cells as cellular immunity because the T cells are going to interact directly with the antigen-bearing particles to remove them from the body, to kill those cells and make sure that they are gone from the body. Okay, so cytokines, the 4 different kinds of cytokines, and then these 3 activities that the cells do. So what are the types of T cells that we can find in the body? Well, there are cytotoxic T cells. These are the T cells that will bind and directly attack cancerous and virus-infected cells by secreting the perforins that we've talked about. There's also a group of T cells that are called helper T cells. And these are going to be the T cells that interact with B cells and move them toward producing antibodies. And the reason that the HIV virus is so particularly virulent is because it will go out and destroy the activity of helper T cells. So we have cytotoxic T cells that are going to do the work right then, helper T cells that are going to interact with B cells, and memory T cells that are produced as the cytotoxic T cells divide. And initially, these memory T cells are inactive. But if a couple of months from now we encounter that same antigen again, and it's found in the body, then when the T cell runs up against that antigen, it will immediately divide, producing cytotoxic T cells and helper T cells, and can then get a very rapid reaction to the threat. Notice that when memory T cells do their thing, they don't have to be activated. They are already activated against that antigen. Okay, so that's the activity of T cells. Cellular immunity. They go right out there and take on the cells that are causing the problems, the virus-infected cells or the cancerous cells. The B cells also have to be activated, but when they're activated, they will produce antibodies, which are these very specialized proteins that bind to antigens. And then I think of them almost as like this big orange flag waving, saying, help destroy the thing that I'm attached to. Quick, get over here and destroy it. So when they bind to the antigen, or when they bind to the antigen, then they're going to be a part of the antigen-bearing particle. When they do that, these antibodies are really going to help stimulate the ability of the body to recognize it as a threat and remove it. So B cells are activated by helper T cells that have been sensitized to the same antigen as the B cells. So the T cell then produces, as part of this activation, the T cell produces cytokines, so more of these chemicals that will have an effect on the target cell. So they produce cytokines, and in response to experiencing the cytokines, the B cell will divide. The clone cells from this division, the daughter cells, will differentiate to become plasma cells and memory B cells. Plasma cells are the cells that do the antibody production. So they produce antibodies. They're going to be antibodies that will bind to the antigen that is found on the antigen-bearing particle or associated with it. And so it produces these antibodies against the antigen and then secretes them into the blood and into other body fluids as well. So depending on where the plasma cell is found will depend where the antibodies wind up. So we find antibodies in the blood, but we also find it in interstitial fluid and in lymph as well. The memory B cells, much like the memory T cells, are initially inactive, but later on, so if we get infected again, if they encounter an antigen that they are sensitized to, they will immediately divide, producing more plasma cells and more memory B cells. So memory B cells will automatically get straight to no helper T cell required. They will automatically get straight to the work of them producing the antibodies against the antigen that it's sensitized to. So in a person who's been living for a while, there can be anywhere from 10 million all the way up to 1 billion different types of B cells, with each one of these types being sensitized to a specific and different antigen. And so once a plasma cell is produced and it starts making antibodies, it can crank out as many as 2,000 of these antibody molecules every second, which is just an astounding number. So if there's 60 seconds in a minute, that's 120,000 antibodies per minute being made by a plasma cell. And of course, an individual plasma cell only makes one type of antibody. But remember, a pathogen, so here's our little bacteria, it may have antigen A, shaped like a little triangle out there, but it may also have antigen B, which is shaped like this, and antigen D, which is shaped like this, one that's shaped like this. So there are many different kind of antigens on the surface of a bacterium or on the surface of a cancerous cell. But each plasma cell type can only make antigens or antibodies against one type of antigen. So when we talk about the immune response, we're usually talking about a polyclonal response. So we make some antigens that are antibodies that will bind to this antigen, some antibodies that will bind to this antigen, some that will bind here to this one, some that will bind here to this one, and so on. So that we are flagging on an individual bacterium or an individual virus-infected cell, we're flagging multiple antigens there as each one of the different plasma cells goes to work producing antibodies. And we describe this immune function as being the humoral defense. Not because it's funny, but because the term humor is a very old school. Think like Shakespearean times. So, the 1500s, 1600s, people talked about the humors of the body. So when we ask somebody if they're in good humor, we're asking them, are they, are all the balance, their humor is balanced? And I forget, there are 4 humors. One is sanguine, that's blood. One is phlegmatic, so think the sort of the snotty fluids. Those are the only 2 I can think of. But there were 4 liquids that were important to remain in balance back in those times, and they were referred to as humors. And because the antibodies are found in body fluids, we talk about this immune function as being humoral defense. It's the defense that's found in the body fluids.